When an animal consumes a specific tasting stimulus, an ingestion is quickly followed by a toxin-induced illness. Future ingestions of the stimulus is less likely. This is typically referred to as a conditioned taste aversion. A conditioned taste aversion involves the avoidance of a certain food following a period of illness after consuming that food. These aversions are a great example of how classical conditioning can result in changes in behaviour, even after only one instance of feeling ill. Conditioned aversions were readily acquired when distinctive flavours are appropriately paired with gastrointestinal distress. Illness-induced aversions have been found to be exceptionally efficient. Long-lasting aversions have been reported in both humans and rats following a single pairing of a distinctive flavour. 3 experiments were undertaken on rats using lithium chloride as an inclusion in order to produce an emetic response, that is, to produce vomiting, when included with alcoholic drinks. The purpose of this is to determine if the induction of the illness associated with the consumption of alcohol can be used to treat alcoholism. The authors aim to test the longevity of the aversion to alcohol to maximise the effectiveness of emetic therapy and to determine if an increased number of pairings produce a lower extinction rate. Also to determine if the strength of the solution ingested has a bearing on the acquisition of the conditioned aversion. It is hypothesized that using emetic aversion therapy with varying alcohol percentages within the samples, the tendency to respond to different but similar stimuli would become a constant. If alcohol were paired with a severe illness, a stronger aversion would be the outcome and a longer-lasting aversion would be achieved. The number of the pairings of the induction of the illness would have no bearing on the effectiveness of the aversion. Cyclochlorophamide, a common source of illness-induced aversion induction within animals, experiments, and is known to produce nausea in humans. In addition to producing aversions having stronger initial magnitudes, larger dosage also produce longer-lasting illness-induced aversions in rats, as assessed by free-choice extinction tests that were initiated soon after the conditioning. The subjects were 108 male, experimentally naive Sprague-Dawley rats that were approximately 90 days old at the beginning of the experiment. The rats were individually housed in commercial wire cages and maintained under a natural light-dark cycle. Except where as noted, the rats had ad lim access to Purina laboratory rodent chowder and food. Different alcohol percentages were used within the samples over a 21-day period with varying intervals of exposure to test if the intervals or concentration has any bearing on the results. The subjects were randomly assigned in one of 16 groups, each containing 6 to 7 rats. All were subjected to 28-hour of fluid deprivation and 4 hours of food deprivation. These deprivation periods terminated at a specific time, which marked the beginning of the access to the solution of sodium saccharide in tap water. This solution was available to each for a maximum of 10 minutes and for at least 5 minutes after the onset of drinking. 5 minutes after saccharin solution removal of the 12 experimental groups received injections of the cyclophosphamide, which was now called cytosine. 4 of the experimental groups received 2.5, which is the low group, 4 between 12.5 milligram per kilo dose, which is the medium, and then the remaining 4 received 25 milligrams, which is the high dose. Rats of 4 control groups were similarly injected with a volume normal saline that was equal to the volume of the maximum dose. 1 saline control group and 3 groups of representative of the different drug dosages were randomly designated as immediate groups. Assessment of aversions acquisition and the rats began 2 days after injection. The standard interval is used in the most taste aversion studies within the laboratory. 3 other randomly determined squads containing 1 saline control and 3 drug groups were then assigned to 20 to 40 day delay groups. These squads were maintained on ad lib food and water and designated delay interval period for the onset of aversion testing. Testing of each rat involved 30 consecutive days of standard 2-bottle preference tests, which separate bottles in saccharid solution. The bottles were weighed daily and refilled when necessary. Bottle positions were alternated as well. They weighed them to determine if there was a preference to either. The design permitted an assessment of aversion degradation as functions for both begetting and extinction. Immediate preference testing revealed that the low gross group produced marginal transitionary taste aversion. Preference scores of the low group exceeded those of the 2 higher dose group, being lower saline control scores. The low dose aversions were highly vulnerable to degradation through extinction. Immediate conditioning for getting also exerted a strong influence on the low dose group. Medium dose groups produced aversions that were initially quite strong and was similar to high dose aversions during all immediate conditioning extinction periods. However, the aversions were not in fact as strong as high dose aversions. Medium dose aversions was significantly smaller than high dose aversions during day the 40-day delay period. These doses produce initial stronger versions of the immediate test condition. However, unlike the low to medium dose group, high dose group showed no aversion degradation as a result of forgetting over the range of the experiment. The results of the experiment strongly support prior arguments that during covert sensitization alcoholism treatment, a major effort should be made to induce intense nausea. Results of the present animal experiment suggest that doses of emetic drugs that are just large enough to produce evidence of effective conditioning during treatment may not necessarily result in a degree of outcome benefit that would be obtained in a larger magnitude doses. The higher the dose leading to the more severe reaction produced the longer lasting results. The number of pairings of the stimulus had no illness, and illness had no bearing on the results or the severity of the illness.